This is the lab where basically everything that we bring to science festivals, science classrooms, gets prepped. What our probably most sought after activity is DNA electrophoresis. We have enough electrophoresis equipment to be out in multiple classrooms, and so each group would get one of these, a group of 3 or 4 students, and would run a DNA fingerprinting type gel. And the way that this works is electricity. First of all, you have a basically medium in here that you pour that becomes a solid. That is a matrix for DNA particles to pass through. You run electricity in here, create an electric field, and DNA, being a charged molecule, will actually move through this medium. And what happens is larger particles move slower than smaller ones, and so you can get a fragment pattern, and each person has different fragment patterns depending on how you manipulate the DNA. So in addition to this kind of activity, this is the really nuts and bolts of what people want to do these days. They want to show students, teachers want to show students some sort of real-life, relevant experience that science can give them. And what we can do with electrophoresis is DNA fingerprinting, or we can amplify a single gene out of an entire genome and look at it using DNA electrophoresis, determine that that gene is there, whether or not that gene is a mutation or not, and things like that. We can also use this equipment for looking at different protein profiles. So for instance, you can actually study an evolutionary type concept, look at different protein profiles of various organisms. So this is definitely the meat of the operation. We have extraction type of kits that go out to the classroom that consist of a solution that's pre-measured and a filtering type system that allows the students to extract the DNA from a certain fruit, or in the case of Festival of Books, we used a different type of extraction solution, and actually this is actually just Dawn dishwashing detergent, and used it to extract the DNA from the kids' cheek cells. So the kids walked out with some of their own DNA in a tube that they make into a little necklace. It's kind of a very fun, engaging activity. Whereas in the classroom, we really emphasize the learning of the cell, how we break the cell open, and how we differentially precipitate DNA away from everything else in the cell, and allow the students to really understand the scientific concept behind DNA extraction.